So drugs can have a lot of different targets. The main targets include the cell wall, plasma membrane, ribosomes, metabolic pathways, and DNA or RNA synthesis. We'll start by talking about the drugs that attack the cell wall. So remember I talked about how peptidoglycan is unique to bacteria. So that's an easy target. And it's easier to target the peptidoglycan in a gram-positive versus a gram-negative. Talked a little bit about this in class, but in a gram-positive bacterial cell wall, the peptidoglycan is all exposed. It's this thick layer on the outermost surface of the cell. Well, in a gram-negative, it's kind of hidden inside here. The antibiotic would have to get through the outer membrane to access the peptidoglycan. So the cell wall targets, the ones that affect peptidoglycan, are usually more effective against gram-positives than gram-negatives. That doesn't mean that they can't work at all against gram-negatives, it's just a lot more effective against the gram-positives. Major categories of cell wall targeting antibiotics include the beta-lactams, penicillin, cephalosporins, the glycopeptides like vancomycin, and then bacitracin. The penicillins, so one of the biggest categories of beta-lactam antibiotics are the penicillins. There's penicillin, which is just one drug, but penicillins as a group includes all of these different drugs. So anything with "-cillin" at the end, you can see all of these different drugs fall under the umbrella of the penicillins. They isolated penicillin from the mold Penicillium notatum, and I really hope you guys got a chance to watch that video. It's like takes you back to grandma's basement, but that is an amazing video that goes through the history of the development of these drugs. So hopefully you guys watched the whole thing. But they discovered the initial penicillin, and then they've semi-synthetic, created all of these different types of drugs from that. The mode of action, it blocks synthesis and repair of peptidoglycan in the cell wall. So as this cell starts to divide, it starts to expand its cell membrane and expand its cell wall to split into two cells. It's like making copies of itself. It can't make new peptidoglycan. So during cell division, the cells just rupture because the cell wall becomes weak and frail, and then it messes with the osmolarity of the cell and it just bursts open. These are effective against gram-positives and some gram-negatives like syphilis, and it's a drug of choice for group B strep. Group B strep, if you've never heard about this, this is a huge one in pregnant women. So like 30 to 40% of the population is naturally a carrier for group B strep. It's a normal skin bacteria, it's not an STD, it doesn't cause any infection, it doesn't have any symptoms on the woman. But if a woman is carrying this while she goes into labor, the woman can pass this to the baby, and the baby can end up getting some very serious infections, including respiratory failure, meningitis, pneumonia. There's all sorts of things that can go wrong with the baby, and the baby can die, and it's a very high risk to the baby. So they'll test the mom for group B strep close to the end of her pregnancy. And they do this by swabbing around the vagina and around the anus, and then they'll send the swab to the lab. They can do this by DNA testing, or they can try to culture it. But once they test positive for group B strep, when the mother's in labor, she has to be hooked up to an IV antibiotic. And then as soon as the baby comes through the birth canal, the baby also has to receive antibiotics. So it's a very serious thing, and women have to be tested for this. Penicillin is one of the drugs of choice for group B strep and the penicillin group. Resistance. Bacteria can produce penicillinases or beta-lactamases that degrade the beta-lactam ring of penicillin. So penicillin has this beta-lactam ring. It's a ring-like structure that can affect the synthesis and repair of peptidoglycan. Some bacteria have developed these little enzymes that can break that apart. So if you put penicillin near it, they secrete the enzymes, and the enzymes chop up the antibiotic, and then obviously the antibiotic is no longer functional. The cephalosporins. So Here you can see cephalosporins, first generation, second generation, third generation, fourth and fifth generation. There can be lots of other generations. So a generation is just talking about a new kind of semi-synthetic model for that type of drug. They're all classified under the cephalosporin group, but you can see they all have different names. And cephalosporins are going to have a very similar mode of action, similar type of resistance. The glycopeptide, vancomycin. Vancomycin is a narrow-spectrum antibiotic. It blocks the synthesis and repair of peptidoglycan in the cell wall, but this one can also affect protein synthesis. So if I'm talking about on an exam or on a quiz, which one can affect the cell wall and protein synthesis? It's vancomycin. Vancomycin is effective against MRSA. It's given to patients with penicillin allergies. So MRSA, methicillin-resistant Staphylococcus aureus. So Staph aureus is a pathogen that causes skin infections, and it can cause boils and weird types of rashes. Usually Staph aureus hides up inside of your nose, and then it can release these exotoxins that can have different effects, and there's all sorts of different diseases it's caused by Staph aureus. There's Staph aureus food poisoning and all sorts of stuff. So it's all because they secrete out these exotoxins. So the bug itself, the microorganism, is hiding in your nose, secreting out toxins that are making you sick everywhere else in your body. When we talk about MRSA, methicillin-resistant Staphylococcus aureus, it's not just a bad one because it happens to be resistant to one of the major penicillin drugs, methicillin. It's hypervirulent as well. So this one pumps out a lot of toxin and can make people really sick. The infections caused by MRSA are extreme infections. So with MRSA, since methicillin doesn't work, you usually give vancomycin. Vancomycin is kind of a last resort drug. So if vancomycin doesn't work, it's really difficult to find another drug to treat. So patients with penicillin allergies also benefit from vancomycin. Resistance. There's a form of vancomycin-resistant Enterococcus that's causing serious issues in the healthcare field. So Enterococcus is a microorganism that lives inside of your digestive tract. And in the digestive tract, it usually doesn't cause any problems. But here's the issue. If Enterococcus becomes resistant to vancomycin, Enterococcus can hand out its gene for vancomycin resistance to other microorganisms. So let's say this really unlucky patient comes to the hospital, and this is like worst case scenario, but it has happened. So a patient comes in with an infection, and they're allergic to penicillin, so they're given vancomycin. So they take the vancomycin, and their primary infection goes away. However, the Enterococcus that was inside of their digestive tract, the ones that were resistant to vancomycin, are now selected for. Because when you put the drug in the presence of them, it kills off the ones that are susceptible, leaving the ones that are resistant. So now you have Enterococcus that are resistant to vancomycin. Now let's say this Enterococcus dislodges somehow, and it comes in contact or in proximity to the person's nasal cavity, where you happen to have methicillin-resistant Staphylococcus aureus. The Enterococcus is going to hand MRSA its vancomycin resistance gene, and then the MRSA will become vancomycin resistant as well. So this is a very serious issue in the healthcare field, and it's pretty prevalent. So you can have this resistance spread from one microorganism to the next. Bacitracin is a narrow-spectrum drug that's found in Neosporin mixed with other antibiotics. This one blocks the synthesis and repair of peptidoglycan in the cell wall. It's effective against skin bacteria, staph, and strep. And resistance is not a serious issue. It's actually used to identify microorganisms in the lab. Later on in the semester, when we start dealing with staph and strep identification, you'll see that we use discs of bacitracin to identify microorganisms.